So for today's class, we will look on toward the classification of the sugars. And among the classification, we have said that there are the monosaccharides and then there are the disaccharides. And today we will see that what these disaccharides are. And as our disaccharide itself indicates that we will have some double sugars or two monosaccharides that would be linked together. And whenever I have to be making these double sugars, so I need to be linking, I need to be condensing two monosaccharides together. When two monosaccharides would be condensed together, so we would make these disaccharides. For the names that must click in your mind for the disaccharides is that we have this maltose coming, we have this sucrose coming, and then we have this lactose coming. So these three are the commonly existing disaccharides that we have got under the category of the classification of the carbohydrates. Now the question is that how come we would be making the maltose, the lactose, and the sucrose? So the simple thing is that if I would make or I would combine two glucose or two monosaccharide with one another, and that too by the elimination of the water molecule, so we would make maltose out of it. If I were to have the glucose and if I would combine it with the galactose, see, this is one monosaccharide, this is second monosaccharide. When linked together, we would make the lactose out of it, and along with that, there would be elimination or the removal of the water molecule. And as of this, if I have to make the sucrose, then one monosaccharide I have to take up is the glucose, the other I have to take up is the fructose with me. When they would be condensed together again, I would end up making the sucrose out of it. If I were to talk about the molecular formulas of this, so all of these disaccharide, their formula goes as C12, H22, and O11. Now the scenario is that if I am combining two monosaccharides together, so what we expect is that their formula should become like 6 combined with 6 should have made 12. 12 hydrogen of one monosaccharide combined with the 12 hydrogen of another monosaccharide should have made 24. And these 6 oxygen when combined with another 6 oxygen of the other monosaccharide, so this should have made it as 12, but this is not the case because I have to take one water molecule out of it. So instead of the disaccharide formula becoming C12H24O12, this is C12H22O11 because two hydrogen and one oxygen. I have removed it in the form of the water molecule. So this is why their formula is same for the maltose, for the sucrose and the lactose. Now again, they are the isomers of one another, although the carbon, hydrogen and oxygen number is same in them, but their structural arrangement is different, so they are given the different name as of their existence is to be bothered about. So the sucrose and the maltose, they would be available within the plants. Okay, but we can consume them, we can use them, we humans can eat them up and we can digest them off. If I were to talk about the animal's milk, so in the animal milk we have got this disaccharide and this is lactose with us. So this is what we are done with the disaccharides. Now would come the third classification of the sugars, and this classification they are entitled as the polysaccharides, or we refer to them as a complex sugar. Poly mean multiple, saccharide means sugar. So when multiple of the monosaccharide are linked together, so we would make these complex sugars. Okay, like I need to have some tens or hundred or thousands of the monosaccharides that would be condensed together by the removal of water when we will link them up together. So we would make them up. Okay, and likewise we must know that where do they exist? So they lie within the plants and they also lie within the animals. Now here you need to know is that what are the names of these complex sugars. So we have starch, then we have the cellulose, and then we have got the glycogen. The starch and the cellulose, they would be available within the plants. The glycogen would be present within the animals. And all of these complex sugars, they are the polymers of the glucose. Like I have to be taking the glucose. Glucose, I would be linking it in multiple ways or in different kind of a configuration. And then lastly, we would end up making all of these. Now the scenario is that the plant, they make starch, we humans can eat it up and we can digest it off. We humans cannot digest the cellulose that is coming from the plants, but this is usually present in the roughages or the fibers that we eat. So it would relieve the problem of the constipation. And we will see in a while that how come this glycogen would be formed. Okay. So as I have written it in term of the note, that the plants they would have the starch and cellulose, animals will have the glycogen. The human can eat the cellulose but can't be digested. The humans can eat the starch but it would be digested by the animals because we have the enzymes in our body that would allow us to be digesting these starch molecules. Now, the scenario is that how come the plants that we have with us so they can make the starch. So you see that here we have got the diagram of the leaf. And earlier in the organization thing, we have discussed about the xylem and the phloem, like in the context of the specialized cells or the tissues that we have in the plant. So over there, I said that the xylem and the phloem, they make the vascular bundles. How come they make the vascular bundles? Because they would be transporting multiple things in the plant. So what usually happens is that the light, it would fall on the leaves. When it is falling on the leaves, so the water would combine with the carbon dioxide gas, and here we have made up the glucose out of it, and there would be release of oxygen gas out of it. Some of the glucose they would remain as it is, but on some of the glucose, some enzymes would work upon, and they would convert this glucose into the fructose. And afterward, this glucose and the fructose, they would link with one another and they would make the sucrose, this sucrose that we have made out of the condensation of the glucose and the fructose. Now this would be loaded into one of the vascular bundle that we have got. So this is the phloem, and this phloem would carry the sucrose to the storage organs. And among the storage organs, we have these root tubers, we have these stem tubers. In this, we would unload the sucrose and they would be linking with one another within these storage organs, and over there we would make the starch out of it. So from the paper point of view, what you must know is that the glucose molecules, when they also could be linked within the chloroplast, and over there some of the starch could be also available. But most of the glucose in the form of the sucrose, by combining the glucose with the fructose, we take them toward the storage organ. So here you know that the sugar that we make out of the photosynthesis, this is glucose with us. The sugar in the form in which it could be transported, so this is sucrose with us. And the sugar that is stored within the plant, so this is entitled as the starch, we say. So this is all we have got in case of like how come the starch could be formed. So by the mechanism of how the starch is being formed, so I would cause the plant to photosynthesize and would allow them to make the glucose. The glucose would combine with the fructose and we would end up making the sucrose. And this sucrose would then be loaded or would be translocated within the phloem vessels. And these phloem vessels, it would carry it to the storage organs, and over there we would convert them into the starch thing. So this is how we have ended up making the starch. Now the other scenario is that in the starch, how come the glucose molecules they would be linked? Okay, since in the plants we have to make sure that in less of the space we should be able to be storing more of the glucose. So I have two choices, or I have got two options to do so. I would take the glucose molecules and I would keep on linking them with one another, and I can arrange them in a spiral manner. The other way could be that I can take the glucose molecules and I would keep on linking them with one another, and I could also add some of the branches to it in the form of the glucose linkages, like they would be attached in the form of the branching scenario. So this is how the starch is being stored in the plants, either in the branching manner or either in the spiral manner. Okay. So the thing is that how is this glucose arranged with the starch? As I've said, so this is a description written that the glucose, they are linked with some attractive bonds, either in the spiral or the branched manner. And the reason for this is that we would be able to pile up more of the glucose within the less given space. The properties we must know for the starch that this is not sweet. This is not soluble. So if this is not soluble, so this is an advantage because it would never ever interfere with the movement of the water molecules. And it can be easily broken down into the glucose molecules whenever there is need of it. Moving onward, if I were to tell you about the cellulose that I have here, so this again is a polymer of the glucose, but the glucose they have to be linked in a different pattern because the cellulose has to make the cell wall, and the cell wall need to have a lot of strength in it so that it do not burst or it should not become elastic, but rather it should remain rigid. So if I cause a chloroplast to make the glucose, and if these glucose molecules, if they are linked in the linear manner or in the form of the straight chains, So ultimately, we would make the cellulose out of it. So if I take the glucose and I link them in a linear manner, so we would end up making this cellulose molecule, and the cellulose is making the cell wall of the plant. Now, lastly, here what we have got with us to be understanding is that how come the glycogen can form within the living organisms. Now, the slight situation is that we people eat more of the glucose than we require for our body. So out of this extra glucose, some of this would be used by the cells, but any of the extra glucose that I would be forming within my body, so it has to be converted into the glycogen, and this glycogen would be stored in the liver of the animals. So as of for the glycogen, we should know that this again is a polymer of glucose, and this again is not sweet in taste, and this again is not soluble with us. Now, if I were to show you the human body, so here you see that at this part of the body, below our neck, we have got the thoracic cavity where the lungs and the heart would lie. And below the diaphragm that we have here, so there would be line up of the organs like the liver and the stomach and the pancreas and the kidneys and the small intestine. So some association is brought about in between the liver and the pancreas that allows the excess glucose to be converted into the glycogen. So you see, here what we have got is a liver with us, and along with the liver, we have got this another organ being placed, and the name of this organ is pancreas. Now, whatsoever the sugars that you have eaten it up, when this would be coming to the small intestines, so in the small intestine, we would end up making or we would be digesting it into the glucose. The glucose would be absorbed into the blood, but when it would come into the blood, the situation is very simple that it would be detected that the amount of the glucose has gone above the limit that was required for the body. So when that is the situation, these pancreas that we have got over here, so they would detect a change in the blood glucose level, and these pancreas they would respond by secreting a hormone out of it, and the name of the hormone is insulin. This insulin would be sent to the liver, and this would also be sent toward the muscle. And what this insulin is going to do that within the liver, it would convert the glucose, excess glucose, not the glucose that is a requirement of the cells, but the excess of the glucose molecules, they would be condensed together, they would again be linked in the branching manner, and we would make glycogen out of it. So this is how the glycogen is being made inside of the human body. So here, if I were to read this for you, so the human, you see that they eat more sugar than is required. And ultimately, these sugars they would be digested off into the small intestines in the form of the glucose molecules. This glucose it would be carried by this vein, which is the hepatic portal vein, and it would carry the glucose from the small intestines, and ultimately it would come into the liver. But here the glucose is in excess. So the solution is that the pancreas that we have got, they would detect an increase in the blood glucose level. And later they would allow the insulin to be excreted out of the pancreas. The insulin would be carried to the liver and the muscle cells, and over there the glucose would be converted into the glycogen. Now again, the thing is that how come these glucose molecules they are arranged within the glycogen molecule? So as is the scenario with the starch where this was packed in the form of the branching manner. So same way for the glycogen, these glucose molecules they would be linked with one another and linked with one another in the branching manner, and we would make the glycogen out of it. But something also happens opposite to it, like in some case, if I was fasting and the glucose level goes below the set limit. So any of the glycogen that we stored earlier, now on this the pancreas would send another hormone. This is glucagon, and the glucagon would convert the glycogen into the glucose molecule. So if I have excess glucose, so by insulin I will convert it into the complex sugar, which is glycogen. And if I have to take the glucose back out of the glycogen, so the pancreas would secrete a hormone out of it, and that is glucagon. So this is all for the carbohydrate section. I hope you would be clear with this. So see you in the next class for the protein lecture.